Hello, Tech House viewers. Welcome to today's video. In this episode, we're going to delve into the world of genetic engineering, specifically focusing on CRISPR technology. We'll explore how this powerful tool is revolutionizing medicine, agriculture, and the environment, and discuss the ethical considerations surrounding its use. So, let's get started. Section 1. What is CRISPR? CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a revolutionary gene editing technology that allows scientists to precisely edit DNA sequences in living organisms. This tool, inspired by a natural defense mechanism found in bacteria, has opened up a world of possibilities in the field of genetic engineering. To understand how CRISPR works, let's first talk about the basics of DNA. DNA is a long, double-stranded molecule that carries the genetic instructions for all living organisms. These instructions are written in a code made up of 4 chemical bases: adenine A, cytosine C, guanine G, and thymine T. The order, or sequence, of these bases determines the information available for building and maintaining an organism. CRISPR technology uses a molecule called RNA, which is similar to DNA, to target specific DNA sequences. The RNA molecule guides an enzyme called Cas9 to the target sequence, where Cas9 then acts like a pair of molecular scissors, cutting the DNA at the specific location. This cut allows scientists to either remove, add, or modify genetic material in a highly precise manner. Section 2. CRISPR in medicine. One of the most promising applications of CRISPR technology is in the field of medicine. Genetic disorders, such as cystic fibrosis, muscular dystrophy, and sickle cell anemia, are caused by mutations in specific genes. With CRISPR, it's now possible to correct these mutations and potentially cure these devastating diseases. One example is the use of CRISPR to treat Leber congenital amaurosis, a rare form of inherited blindness. In 2020, researchers reported the first successful use of CRISPR to treat this condition in a patient, marking a significant milestone in the field of gene therapy. CRISPR is also being explored as a potential treatment for cancer. By editing the immune cells of patients, scientists aim to boost their ability to recognize and attack cancer cells, potentially offering a powerful new weapon in the fight against this deadly disease. Additionally, CRISPR holds promise for tackling viral infections, such as HIV and hepatitis B. Researchers are working on using CRISPR to remove viral DNA from infected cells or to disable the genes that allow viruses to replicate, which could lead to new and more effective antiviral therapies. Section 3. CRISPR in agriculture. CRISPR technology is also revolutionizing agriculture by enabling the development of genetically modified crops with desirable traits, such as improved yield, resistance to pests, and tolerance to drought or other environmental stresses. For example, scientists have used CRISPR to create rice plants that can better withstand flooding, a major challenge for rice farmers in many parts of the world. Similarly, researchers have developed tomatoes with an extended shelf life, which could help reduce food waste. Another application of CRISPR in agriculture is the development of crops with enhanced nutritional content. Scientists have successfully used CRISPR to increase the levels of essential nutrients, such as vitamins and minerals, in staple crops like rice and wheat. These genetically modified crops have the potential to address malnutrition and improve global food security, particularly in developing countries where access to a diverse and balanced diet may be limited. In addition to modifying plants, CRISPR is also being utilized to engineer livestock for improved health and productivity. For example, scientists have used CRISPR to create pigs that are resistant to a common and costly viral disease called porcine reproductive and respiratory syndrome (PRRS). By enhancing the health and well-being of livestock, farmers can produce more food while reducing the need for antibiotics and other treatments. Section 4. CRISPR and the environment. Beyond medicine and agriculture, CRISPR technology holds promise for addressing various environmental challenges. One such application is in the field of conservation, where CRISPR can be used to edit the genes of endangered species, making them more resilient to threats like disease, climate change, and habitat loss. For instance, researchers are investigating the potential of using CRISPR to modify the genes of coral species, making them more resistant to the bleaching events caused by rising ocean temperatures. By enhancing the resilience of these vital marine ecosystems, scientists hope to mitigate the devastating impacts of climate change on coral reefs and the countless species that depend on them. Another environmental application of CRISPR is in the area of pest control. Scientists are working on using CRISPR to engineer sterile or non-reproducing insects, such as mosquitoes, which could help control the spread of diseases like malaria, dengue, and Zika virus. By releasing these modified insects into the wild, populations of disease-carrying pests could be significantly reduced, potentially saving millions of lives. Section 5. Ethical considerations. While CRISPR technology offers numerous benefits, it also raises important ethical questions. One of the most debated concerns is the potential for designer babies, in which parents could use CRISPR to select specific traits for their children, such as intelligence, appearance, or athletic ability. This raises concerns about social inequality, as only those who can afford such procedures may have access to these enhancements. Another ethical issue is the potential for unintended consequences, as the long-term effects of CRISPR gene editing are not yet fully understood. Editing the genes of one organism may have unforeseen impacts on the broader ecosystem, and there is a risk of off-target effects, where CRISPR unintentionally edits other genes, potentially causing harm. Lastly, the possibility of using CRISPR for malicious purposes, such as creating bioweapons or engineering invasive species, has raised concerns about the need for appropriate regulation and oversight of this powerful technology. Conclusion: CRISPR has undoubtedly revolutionized the field of genetic engineering, offering the potential to transform medicine, agriculture, and the environment. However, as with any groundbreaking technology, it is crucial to carefully consider the ethical implications and ensure that its use is guided by responsible policies and regulations. Thank you for joining us in this exploration of CRISPR and the future of genetic engineering. If you found this video informative and thought-provoking, please be sure to like, share, and subscribe for more content on cutting-edge science and technology. Until next time.